the success of any initiative. The SARS-CoV-2 pandemic, declared as a global health emergency by the World Health Organization, appears to be substantially exceeding, creating a devastating crisis that may leave deeply wounded scars on the way we perceive our world and everyday lives. The contagion's extremely high potential for dissemination and the development of different variants has taken toll on lives worldwide. Today, the PG Department of Zoology is organizing an international webinar series on dynamics of immune response to SARS-CoV-2 and other human coronavirus in COVID-19 under the initiative Moving Beyond Horizons to Dream, to Explore, and to Discover. With all reverence and due respect, I would like to welcome Dr. Sister Gisela George, the Principal, Mercy College, Palakkad, the most esteemed chief guest of the day, Dr. Rashmi Verma, Senior Scientist, Clinical Proteomics Core Lab, Department of Pathology, University of California, USA, Dr. S. Jayashree, Head PG Department of Zoology, respected teachers, and all young learners attending the session online. It gives me immense pleasure to welcome you all to this session, to this program. To begin with the session, let me invite Dr. S. Jayashree, Head Department of Zoology, to address the attendees online and introduce the guest of honor.
Respected dignitaries on the online platform, Dr. Rashmi Verma, Sister Principal, Mercy College, and dignitaries from other institutes abroad, from India, and my dear colleagues and dear students, good morning, good noon, and good evening. Let me the chief guest of the day, Rashmi Verma. Dr. Rashmi Verma earned her PhD from Sri Chitra Tirunal Institute of Medical Sciences and Technology, Thiruvananthapuram, and subsequently joined for postdoctoral research at the University of California, Davis, under eminent professors like John Crew and Paul Lucif, who is the first one to clone the HIV genome. Dr. Verma holds a senior staff scientist position at Clinical Proteomics Core Lab, California Davis Medical Center. She has a broad background in infectious pathogen immunology with specific efficient and high throughput multiplex approaches for disease-related immune biomarker studies. Dr. Verma is the key researcher in several collaborative national and international infectious disease research with scientists and clinicians in the US, India, Uganda, and Pakistan for the chronic lung infectious disease tuberculosis. Published in peer journals, including Nature. So, all of us have gathered here on this online platform to hear from you, madam. And we are also on a deep brisk of COVID-19 pandemic. So, on behalf of the entire participants on this online platform and on behalf of PG Department of Zoology, Mercy College, welcome you to deliver the keynote speaker on dynamics of immune response to SARS, coronavirus 2, and other human coronavirus COVID-19 pandemic. Once again, with pleasure, inviting and welcoming you. Okay.
Thank you for the introduction and good morning, everyone. And I would like to thank Dr. Jayashree for inviting me to present my work today. So let me start sharing my screen. So can you all see my screen?
Yes.
Okay, so today we will be discussing about the dynamics of immune response to SARS-CoV-2 and other human coronaviruses in COVID-19 patients. So the picture on the left here shows Professor Charles Rice, who is the 2020 Nobel recipient for physiology and medicine for his discovery on hepatitis C virus. He's an alumni from my university, so I just want to share that glad news with all of you. So for those who are not familiar with basic immunology, couple of basic immunological concepts. What is antigen and antibody? So antigen is a substance against which our body elicits an immune response. So an antigen can be bacteria, viruses such as SARS-CoV-2, proteins, polysaccharides, pollen, and so on. Antibodies are molecules produced by our body to fight off the antigen. So antibodies are basically proteins. There are several classes of antibodies, but the two major classes are called IgM and IgG antibodies. IgMs are the primary antibodies which the body makes when it encounters an antigen for the first time. IgMs are not specific to the antigen, so body makes them right away to start off the fight, while it takes time to make IgG antibodies, and IgGs are specific to the antigen, so that's why body needs time to make them. So usually it takes 7 to 21 days for the body to make IgG antibodies against a pathogen. So once IgGs are made, IgGs take over the job from IgM, and ultimately IgGs neutralize the antigen and macrophages destroys the antigen. So that's the basic immunological concept. And how do you measure antigen-antibody interaction in a clinical lab? So there are several techniques to do that. Most common technique is called enzyme-linked immunosorbent assay, abbreviated as ELISA. So the basic principle of antigen-antibody interaction is like lock and key concept. You can consider antigen as the key and antibody as the lock, and the key fits snugly into the lock. So ELISA takes advantage of that principle. So the technique that I use to measure immune response to SARS-CoV-2 and other coronaviruses is by a modified ELISA technique. It's called multiplex microbead antibody assay. It's a high throughput ELISA. So I'll describe how that works in the subsequent slides. So a brief overview of the topics that we will be discussing today. So I'll talk about what multiplex antibody assay is and how it differs from traditional ELISAs. And whenever we do assay development, we optimize the assay using animal models if available. So I'll talk about our animal models for COVID-19. Once the assay is optimized using animal models, we move on to test human samples. So we test healthy controls as well as COVID-19 patients against 7 coronaviruses. So we also did a time course study where we collected longitudinal samples from several COVID-19 patients, ranging from few days to several weeks post symptoms, and we measured the immune response in these patients. So in addition to antibodies, we also measured several cytokine chemokines and growth factors in these patients. I'll talk about that. And finally, immune response in vaccinated people, which I guess most of you want to know. So what is multiplex microbead immunoassay and how it differs from ELISA? Multiplex microbead immunoassay is abbreviated as MMIA. So I guess most of you are familiar with how ELISA works. Therefore, I'll not go into any of the details, but briefly, in an ELISA, analytes are bound to the wells of a polystyrene microplate or 96 well microplate by passive adsorption. Analyte can be either antigen or antibody, depending upon the type of the reaction. So in this talk, when I say analyte, I'm talking about protein ELISAs, not carbohydrate or something else. Because the immune response that we looked at in our assay is for SARS-CoV-2 proteins. So in MMIA, analytes are bound to the surface of a microbead by chemical cross-linking. These beads are the size of a red blood cell, about 6 to 8 micrometers in diameter. These beads are carboxylated on their surface. And as you know, proteins have amino groups. So what happens is amino group of the proteins react to carboxyl groups on the bead surface with the help of 2 chemicals. So in ELISA, analytes are attached to a microplate by passive adsorption, whereas in MMIA, analytes are bound to the bead surface by chemical cross-linking. So here I show an example of a sandwich ELISA. So what sandwich ELISA is, antigen is sandwiched between 2 antibodies, a capture antibody and a detection antibody. So capture antibody is coated onto the surface of a microplate in ELISA, whereas it's chemically cross-linked to a bead surface in MMIA. Next, we add the test sample containing the antigen of interest, and the capture antibody captures the antigen. This is followed by the addition of a detection antibody, which is directed to another epitope on the antigenic surface. So for those people who don't know what an epitope is, it's a short stretch of amino acid where the antibody binds. So detection antibody binds to another antigenic epitope. In ELISA, detection antibody is conjugated to an enzyme, which in most cases is horseradish peroxidase. We add the substrate for the enzyme, and the enzyme makes a colored product. The intensity of the color can be measured using an ELISA or a microplate reader. Output is called optical density. In MMIA, detection antibody is attached to a fluorochrome, which is phycoerythrin. And the fluorescence intensity is measured using a luminous flow cytometer. Output is called median fluorescence intensity, which is similar to optical density in an ELISA. So that is the technique. So ideally, principle of MMIA is same as ELISA, but it is fluorescence based. So what is multiplexing? What does the term multiplexing means? Multiplexing is the ability to detect multiple analytes in the same sample in one reaction. So how is it possible with microbeads? So what happens is that 2 dyes, fluorescent dyes of red and orange intensities are mixed in various ratios to make 100 uniquely color-coded microbeads, each with a unique internal fluorescence. And the powerful lasers in the luminous flow cytometer are able to distinguish each bead based on its unique internal fluorescence. So the advantage is that you can attach 100 different antigens to each of these beads, mix them together, and add a patient sample and look at 100 different analytes in one reaction in a single sample. So it's extremely high throughput, whereas if you do an ELISA, you would end up doing 100 different ELISAs if you were to look at 100 analytes in a patient sample. So this image shows how beads pass through the lasers. A precision fluidics align the beads as a single file, and each bead passes through the lasers one at a time. Green laser identifies the phycoerythrin fluorescence, caused by the antigen antibody binding on the bead surface, and red laser identifies the bead internal fluorescence. So the image here shows the traditional Luminex flow cytometer. You can see the platform here where the plate goes in, and here is the needle probe which picks up sample from each well. So MagPix is a miniature version of the traditional Luminex flow cytometer. This is a small compact version. So advantage of MagPix is that this machine can be placed inside a biosafety cabinet. So whenever we run assays that requires handling of hazardous samples, this is a very useful model because we can perform the assay from start to finish inside the biosafety cabinet. Whereas if you use a traditional flow cytometer, you still perform the assay inside a biosafety cabinet, but in order to read the plate, you need to bring it out of the BSC. So in summary, MMIA is an ELISA-based assay using flow cytometer technology. MMIA also uses 96-well microtiter format similar to ELISA, but the use is completely different because in an ELISA, actual reaction takes place on the surface of a 96-well plate, whereas in MMIA, reaction takes place on a bead surface, and 96-well plate is used just as a carrier to hold the beads. And as I mentioned, MMIA uses microbeads as solid state for assays, whereas ELISA uses the actual 96-well plate. And since the beads are uniquely color-coded, this allows multiplexing of up to 100 analytes per well. And now there are even modified versions of Luminex flow cytometer where you can multiplex up to 500 analytes. And for protein antigens, we are not there yet, but for nucleic acid, people are multiplexing more than 100. And each individual analyte can be identified using its unique color-coded bead ID. An analyte binding reaction is measured by the amount of phycoerythrin bound to each bead. This allows simultaneous analysis of 100 analytes per sample in one reaction. And MMIA is faster compared to ELISA because ELISA requires an overnight antigen coating and another half a day to finish the reaction, whereas MMIA takes half a day to finish the entire reaction. And MMIA is much more sensitive than ELISA because of the powerful lasers. The dynamic range is much wider. So this is the technology. So I can stop for a few minutes if you guys have any questions about the technique. If not, I can continue. So I think I can continue. Okay, moving on to the topic. So what are coronaviruses? Coronaviruses are a large family of viruses which cause mild to moderate or severe upper respiratory tract illnesses in human. So there are 7 coronaviruses which commonly infect humans. I shouldn't use the word commonly for 7 of them. Actually, out of these 7, 4 of them are called common coronaviruses. They are named 229E, NL63, OC43, and HKU1. These are the common coronaviruses, and these cause mild to moderate respiratory illnesses such as common cold. And most of us are infected with one or more of these coronaviruses at some point in our lives. And many times the infections are so mild that we don't even know that we got the infection and it cures on its own. Over the past 2 decades, 3 new coronaviruses emerged from animal reservoirs, causing widespread devastation and illness in our population. And these are SARS-CoV, Middle East Respiratory Syndrome or MERS coronavirus, and finally SARS-CoV-2. A brief overview of the structure of SARS-CoV-2. 4 structural proteins, spike protein, nucleocapsid protein, membrane glycoprotein, and envelope proteins are essential for the virion assembly and infection of SARS-CoV-2. Spike protein consists of two subunits, S1 and S2. S1 contains a stretch of amino acids called receptor binding domain, abbreviated as RBD, which binds the ACE2 receptors on the host cell surface, helping the virus infect the host cell. So this talk is mainly focused on the spike protein, especially spike RBD, and the nucleocapsid protein. Commercial assays for SARS-CoV-2 detection either look at the viral RNA or viral antigen, or look at the preponderance of IgG or Igm antibodies, which are produced as a result of infection in infected individuals or suspects. So here, for immune response, we look at IgG and Igm antibodies. Therefore, we will not talk about any molecular assays in today's discussion. So this slide gives an overview of how we designed the MMIA for coronaviruses. So you can see the list of viruses in our panel. So we have 7 coronaviruses, SARS-CoV-2, SARS-CoV, MERS, and the 4 common coronaviruses. So I showed different color-coded bead for each of these viruses. So we attached the antigens from each of these viruses to uniquely color-coded beads. And since the machine is capable of identifying these beads by the unique color code, we can mix all these beads together in one reaction. So when I say one reaction, it's a single well in a 96 well plate. So we add all the beads together, and each bead has a particular viral antigen attached to it. Next, we add the test sample. So in this case, it's a serum or plasma from a COVID-19 patient. So if the patient has antibodies, it will bind to the antigen on the corresponding bead surface. And since these are human samples, we are measuring IgG or IgM. So these are human antibodies, right? So how do we detect? We add an anti-human IgG attached to phycoerythrin. So wherever there is human IgG, anti-human IgG is going to bind, and in turn, which is attached to phycoerythrin, right? So this gives the fluorescence where there is antigen antibody, secondary antibody binding. So then the beads are read through the Luminex flow cytometer, and median fluorescence intensity is measured. So if there is a positive signal, we get high median fluorescence intensity. If the sample doesn't contain antibodies, median fluorescence intensity will be very low. So moving on to the data, I mentioned earlier that whenever we develop assays, we optimize the assays using animal models if available. So we used a monkey model for SARS-CoV-2 in our study. So this is a heat map which shows the median fluorescent intensity data in our multiplex assay for monkeys. So right side, you see the color scale, red indicating strong median fluorescent intensity, which means very strong antibody levels, and green indicating low to no antibodies or low median fluorescence intensity. So you can see the list of the antigens in our panel. For SARS-CoV-2, we use RBD of the spike protein, abbreviated as SRBD. So what that means is we have a bead with SRBD from SARS-CoV-2 attached to it. For SARS-CoV, we use 2 beads. One is the full-length spike protein, and second one is nucleocapsid protein. For MERS coronavirus, we use SRBD. For the common coronaviruses, we use full-length spike protein. So these are essentially beads attached to proteins from these viruses, and we mix them all together, add the test sample, then add the secondary antibody attached to phycoerythrin and measure the fluorescent intensities. So you can see two groups of healthy monkeys here. We tested healthy monkeys from our own campus because we have a primate center associated with my university. It's a California National Primate Research Center, which is one of the 7 primate research centers in US. We also use another set of healthy monkeys from Charles River Laboratories. So what you see here is in our healthy monkeys, you don't see any antibodies to any of these viruses, because if the antibody reactivity is strong, you would see a red color, not green color. And you can see COVID-19 monkeys on the top. These are experimentally infected animals. Very strong antibodies to SARS-CoV-2 SRBD is seen in COVID-19 monkeys. So this indicates that the assay is working the way it's supposed to. We also used a rabbit model for SARS-CoV infection. So the graph on the left here shows the data on rabbits. So here I only show the data for SARS-CoV-2 SRBD and not to other coronaviruses. And I show part of the monkey data here too, which I've just shown in the heat map. So let me go over the monkey data first. So this axis shows the median fluorescent intensity, which is abbreviated as MFI. For SARS-CoV-2 SRBD IgG antibodies. So in healthy monkeys, as you have seen in the heat map, you don't see any antibodies to SARS-CoV-2 SRBD, whereas in COVID-19 monkeys, very strong antibodies are seen. And the results are similar in rabbits too. Healthy rabbits have no antibodies to SARS-CoV-2 SRBD, whereas infected rabbits have very strong antibodies to SARS-CoV-2 SRBD seen. We also used a hamster or guinea pig model for SARS-CoV-2 infection. We had 2 groups of hamsters. One group received low dose of the virus and another group received high dose of the virus. And we collected longitudinal samples ranging from 2 to 16 days post-inoculation. Sorry, let me go back. And we also had a group of healthy hamsters. So as expected, healthy hamsters did not have any antibodies to SARS-CoV-2. And in infected hamsters, antibodies appear 7 days post-inoculation, and they stayed very high up until 16 days, after which we did not keep these animals. So in 2 and 4 days, you don't see antibodies coming up. And for high dose hamsters also, the pattern of antibody response was similar to what we saw in low dose hamsters. So ideally for antibody response, we did not find any difference between high and low dose hamsters. So once the assay is optimized in animals, we moved on to human cohorts. So here, this is a heat map which shows the multiplex antibody data in our human cohorts. So similar to the monkey heat map, here is the color scale. Red indicates high reactivity or high antibody levels. Green indicates low antibody levels. And here are the antigens in our panel. So we tested two groups of healthy. One set of healthy individuals are from USA, and another set obtained from our collaborators in Pakistan. In fact, these are pre-pandemic healthy controls. And we've tested COVID-19 patients, both from Pakistan and USA. We have collaborators in Pakistan, so we received a lot of samples from them. So what you can see here is in US healthy, you don't see any antibodies to SARS-CoV-2 SRBD and SARS-CoV and MERS, whereas you can see a lot of antibodies to the common coronaviruses, which is expected because, as I mentioned earlier, most of us are infected with one or more of these viruses in some point in our life. So it's not surprising to see antibodies to these coronaviruses. And Pakistani healthy also had similar pattern as we have seen in US healthy. One difference we noticed between US healthy and Pakistani healthy is that levels of antibodies to one of the common coronaviruses, OC43, found to be elevated in Pakistani healthy. We don't know what that means, but that was an observation in our study. And in COVID-19 patients, both from US and Pakistan, very strong reactivity to SARS-CoV-2 SRBD is seen, and some reactivity to SARS-CoV-S and very strong reactivity to SARS-CoV nucleocapsid protein is seen. So I'll come to this point in the subsequent slides. So in addition to healthy controls, we measured the background reactivity of our assay in disease controls. So what are disease controls? So these are patients who are infected with respiratory viruses other than SARS-CoV-2. So in addition to the healthy, we've tested samples from chronic obstructive pulmonary disease patients, tuberculosis patients, patients infected with respiratory syncytia virus, and influenza patients. And the dotted lines here indicates the assay cutoff, which is the value above which a sample is considered positive. So as you can see, you don't see any antibodies to SARS-CoV-2 SRBD in healthy or disease controls, whereas very strong antibodies in COVID-19 patients, both from US and Pakistan. So here are the results of the heat map in detail. So you see 7 coronaviruses here that I had shown in the heat map for human. So healthy controls from US are shown here, and healthy controls from Pakistan is shown on the right here. So the pattern of antibody reactivity between US and Pakistan remained similar for healthy controls. So in the US healthy, you don't see any antibodies to SARS-CoV-2 SRBD, SARS-CoV, or MERS, whereas all these healthy had antibodies to common coronaviruses. In the Pakistan healthy, no antibodies to SARS-CoV-2 SRBD is seen. You can see one healthy with very high antibodies to SARS-CoV-S protein, and several of Pakistani healthy had antibodies to MERS. And we think this is real and not coming from any cross reactivity because of the close proximity to the Middle East. Perhaps these individuals are infected with MERS at some point in their lives and the antibodies are showing up. So here are the antibody results in COVID-19 patients from US. Very strong antibodies to SARS-CoV-2 SRBD is seen in all 10 COVID-19 patients here. Some cross reactivity to SARS-CoV-S, which are the blue dots here. I think my mouse disappeared. Okay, got it. And very strong cross reactivity to SARS-CoV nucleocapsid protein is seen in all 10 COVID-19 patients from US. This is not surprising because the spike proteins of both SARS-CoV-2 and SARS-CoV-1 share about 70% amino acid identity, whereas the nucleocapsid proteins of both viruses share about 90% amino acid identity. So that's why you see very strong cross reactivity between the spike of SRBD and nucleocapsid. And the pattern of antibodies to common coronaviruses in COVID-19 patients are similar to what we saw in healthy controls. And graph on the far right here shows the antibody reactivity in COVID-19 patients from Pakistan. We have obtained 43 COVID-19 samples from Pakistan. So out of 43, 3 patients did not have any antibodies to SARS-CoV-2 SRBD, and the sensitivity was 94% in this group. And almost all the patients cross-reacted to SARS-CoV-2 nucleocapsid protein in its group, similar to what we saw in COVID-19 from US. We also did a time course study where we collected longitudinal samples from 6 COVID-19 patients over a period of several days to weeks post-symptoms. We measured Igm antibodies, total IgG, and IgG subclass in these patients. So IgG has 4 main subclasses, IgG 1, 2, 3, and 4. So in most of the viral infections, IgG 1 and 3 are elevated, whereas in bacterial infections, it's mainly IgG 2 and 4 which are elevated. So total IgG is shown in black, IgM is shown in red, IgG1 in blue, and IgG3 in green here. So the earliest time that antibodies appear in these patients is about 5 days post symptoms, as you can see in the patient on the far right here, RIP 0020. And this seemed to vary from patient to patient, but in general, most of the patients had antibodies or IgG antibodies about 10 days post symptoms. And IgM also appeared about the same time as the IgG antibodies. And 4 out of 6 patients had IgG1 and IgG3 elevated. I had mentioned earlier that we measured several cytokine chemokines in these patients. And in fact, many of them were of interest, but especially one particular cytokine, it's called CXCL10, another name is IP10, seemed to be of particular interest in these patients. So we did a time course study in 6 patients. And these are the same 6 patients in the previous slide. So what we noticed is CXCL10 was found to be elevated in all 6 patients, but over time, the levels of the cytokine dropped precipitously in all 6 patients, as you can see here. So the graph on the left shows the antibody data in the same patient group. So what you want to see here is as the antibody levels go up, CXCL10 levels drop. So what we speculate here is as the patient health improves, CXCL10 levels drop. So this CXCL10 can be used as a surrogate marker for improved patient health in COVID-19 patients. So we also measured CXCL10 in our Pakistani COVID-19 patients. So out of the 43 COVID-19 patients, we stratified them based on disease severity. So out of 43, we had 20 severe COVID-19 patients. So these patients ended up in ICU and they needed intubation. And 23 were mild to moderate COVID-19 patients. So these patients did not need to be intubated. So here are the CXCL10 levels, and green is the CXCL10 level in healthy individuals. So you can see that the CXCL10 levels are elevated in both severe and mild to moderate COVID-19 patients compared to healthy controls. These are significantly elevated. But between these 2 groups, there was the results were not statistically significant, even though the median line appeared to be higher in severe COVID-19 patients. And the graph on the left here shows the IgG antibodies to SARS-CoV-2 SRBD in the same patient groups. So same observation here, compared to healthy, antibody levels were highly elevated in severe and mild to moderate COVID-19 patients. But between these 2 groups, there was no statistical significance. So moving on to the last part of the talk, vaccine studies. So we had 2 monkey models for vaccine studies. One set of monkeys received an adenovector vaccine expressing SARS-CoV-2 SRBD. And we had another group of monkeys who received adeno vaccine expressing SARS-CoV-2 nucleocapsid protein. So here are the antibody results. So we had 7 monkeys in SARS-CoV-2 SRBD group. You can see that the antibodies are very high in SARS-CoV-2 SRBD vaccinated monkeys, whereas and antibodies to nucleocapsid protein was negative in this monkey cohort. Whereas in the second cohort who received SARS-CoV-2 nucleocapsid, antibodies to nucleocapsid was highly elevated, whereas no antibodies to SARS-CoV-2 SRBD is seen. In fact, our assay can distinguish vaccination from infection because most of the vaccines currently available are spike protein based. So in infected individuals, you expect to see antibodies to both spike protein and nucleocapsid protein, whereas in vaccinated individuals, you only expect to see antibodies to spike protein. So this is the final slide, which I guess most of you want to know, the IgG levels in vaccinated individuals. So in US, 2 main vaccines that we are getting are mRNA-based from Moderna and Pfizer. So here are the antibody levels in Moderna vaccinates. We did a time course study here. We collected samples before vaccination, which is the pre-vaccinated group here. And then later, we collected samples 2 weeks, 4 weeks, and 6 weeks post-vaccination. And here are the IgG levels to SARS-CoV-2 SRBD. So as you can see, in 2 weeks, antibodies start appearing, and they increase in 4 weeks. By 6 weeks, antibody levels are saturated or elevated. And for Moderna, booster doses at 4 weeks. So the 6 weeks time point is in fact 2 weeks after the booster shot. So what you see here is you really need the booster shot to have enough antibodies for protection. So here I show the antibodies to nucleocapsid protein in the same individuals, same Moderna vaccinate. So as I mentioned earlier, in vaccinated individual, you don't expect to see antibodies to nucleocapsid protein. So hence, these individuals are negative for antibodies to nucleocapsid protein. So here are the SARS-CoV-2 SRBD IgG antibodies in Pfizer vaccinate. You see similar pattern as in Moderna. So for Pfizer, booster dose is at 3 weeks. And then this 5-week point is in fact 2 weeks after the booster shot. So as you can see, after the booster shot, antibodies are highly elevated. But to summarize, we have developed a multiplex antibody immunoassay for detection of antibody response against 7 coronaviruses, SARS, SARS-CoV-2, MERS, and 4 common coronaviruses. We have validated the assay in animal models and COVID-19 patients and healthy controls. Earliest time point that the IgG antibodies start appearing is about 5 days post symptoms. Igm also start approximately the same time as the IgG antibodies. Antibodies against SARS-CoV-2 spike and nucleocapsid are SARS-CoV spike and nucleocapsid are cross-reactive to SARS-CoV-2, and little to no cross-reactivity against MERS and other common coronaviruses. And finally, CXCL10 negatively correlate with antibody response in COVID-19 patients, and strong levels of SARS-CoV-2 SRBD IgG antibodies are seen in Pfizer and Moderna vaccinated individuals. So future studies, we already collected 100 from our Pakistani collaborators, 150 COVID-19 patients with severe disease, 150 with mild COVID-19, and 150 suspects who are PCR negative for COVID-19. So the idea here is to collect clinical and immunological data and use analysis, large predictive modeling analysis, computational analysis platforms such as Milo, which is machine integrated operator learning platforms, whether we can predict who will end up in ICU or not. We are collaborating with California National Primate Research Center for COVID-19 vaccine studies in a pediatric monkey model. We also obtained longitudinal plasma samples and PBMCs, which are peripheral blood mononuclear cells from Stony Brook Hospital in New York. So we will be measuring cytokine chemokines. Then we'll be doing RNA-seq. So it's basically genomics, proteomics, and lipidomics in these patients in a time course study. And these are my collaborators. I thank all of them and thank you all for listening.
Thank you, ma'am. Thank you, ma'am, for enlightening us on the critical progression and immunopathogenesis of COVID-19. It was indeed a session with some potentially useful takeaways. Ma'am, now we have some questions in the chat box. Can I read that?
Absolutely, yes.
First, I have a question.
Yes, ma'am.
Actually, I cannot hear you.
Okay, sorry. Is it audible? Is it audible?
Yes.
Yes. So, taken Covishield, and after 2 doses, the antibody titer after vaccination, the antibody titer varies among individuals, and few are having very less antibody titer. So, is they want to take another dose, or why is that is so?
So it's like person to person difference, right? Not all of us behave the same. But I think getting a third dose is still under discussion. Even here for Pfizer, there are some talks on giving a third dose, but I think it's put on a hold. We need more studies. Yeah, I think unless and until we have more data, we cannot decide on a third dose. Maybe these individuals have low immune response, so no matter if they receive a third dose or not, their immune system is like that, perhaps.
Okay, thank you.
Ma'am, our first question was, why can't we recommend for a test dose of any COVID vaccine 30 minutes before giving whole dose to avoid unnecessary allergic reactions?
It's like if you think of the allergic reaction, just imagine the percentage of people who have really severe allergies, right? It's negligible compared to the millions of people who got the dose. And then see these allergies, I think we are overthinking about the allergies. It's not a big deal. You expect that from any vaccine, right? So why make a big deal? And another thing we need to think is, even to get the proper dose, people are standing in line. So are you going to add the waiting time to get the actual shot? So it's all adding time.
Yes, ma'am. Next question. Among dead and live virus, which is better?
Better for what? What's the purpose?
I think, ma'am, for making vaccine like a dead virus vaccine or live.
Actually, for SARS-CoV-2, none of the vaccines use live or dead virus. Most of the vaccines currently available are protein-based.
Okay, ma'am. Next question. Among Keralites, there is a positive swing towards Covishield than Covaxin. Do you think attenuated vaccine like Covaxin could have some negative impact as rumored?
Actually, I'm not sure if there are any proper studies out there which compares both these vaccines. I don't think I've come across any study, so I cannot comment on that. And actually, I just know these brand names, but which one is AstraZeneca? Is it Covaxin or Covishield?
People are choosing more Covishield. There is some kind of rumor about Covaxin.
So is Covaxin the one that's made by Bharat Biotech? Is that the one? Which one is the Oxford one? Is it Covaxin or Covishield? Because I don't know these brand names. Yeah, so unless and until there is any comparative published studies, I cannot comment on which one is better or worse.
Covaxin is Bharat made by, and Covishield is the Oxford one.
Okay, yeah. So as you know, we have more data globally for the Oxford one than the other one, right? So we need comparative studies, and I'm sure someone is doing those studies. So let's wait for a publication to come out.
Okay. Next one, what is the longevity of antibodies in a body of a vaccinated individual? Does it change with their age and health?
So longevity means, so you expect antibodies, at least based on the current publications, for several months, these antibodies are present in the body. And as with any infection, if there is no active infection, it is not necessary for the body to constantly make antibodies, right? So that's why we have memory cells in our system. So what happens is when you get a vaccine, you will have antibodies for a few months, and then the body knows that it doesn't need to make those antibodies constantly. So then the antibody levels, they slowly wane off, but that doesn't mean that your protection disappeared. So when you get an infection next time with the same agent, the memory cells immediately make antibodies. So that's the case with any infection. So you don't need to worry after 6 months or 1 year when you measure the antibodies, and if you don't see antibodies, that doesn't mean that the protection disappeared.
Thank you, ma'am. Next question. Is there any scientific evidence about the gender disparity in the morbidity and mortality of COVID patients in the light of differential expression of ACE2 in male and female?
That's a good question. I haven't really looked for it. I'm sure there are studies out there, because in most of the studies, they disclose the gender. But personally, I haven't really searched for it, but that's a good question.
That was our last question in the chat box. Is there anyone who wants to ask any doubt? Okay, so now we can conclude. Now to conclude, let me invite Ms. Tesna to propose the vote of thanks.
Good morning. It is often felt that there is a tremendous knowledge gap between clinicians, researchers, and commoners dealing with emerging issues of health diagnosis. As a stride towards bridging this gap, Dr. Rashmi, the respected spokesman of the day, had done phenomenal justice to the topic titled by literally covering all relevant aspects critical towards addressing clinical spectrum of COVID-19. Thank you, ma'am, for being with us. We owe our gratitude to the scientific community, whom upon exhaustive investigations are trying hard to unravel the cryptic contagion. Their valuable contributions have helped to delineate certain major challenges in regard to designing of increasingly effective diagnostic, therapeutic, and prophylactic strategies. I would like to thank our principal, Dr. Sister Gisela George, for presiding over the meet. And also, I would like to compliment the PG Department of Zoology, headed by Dr. S. Jayasri, for bringing up the session with some potentially useful takeaways. Finally, with all love and sense of respect, I would like to thank all attendees of the online meet for their kind cooperation and sincere involvement. Have a great day ahead. Thank you.
A humble request from the part of organizing team. Let us all open our cameras to have a photo being clicked with the resource person. Please open your camera.
You can get leave.
Please open your camera. Undala. Thank you all.
Thank you, ma'am.
Hello.
Thank you, Rashmi.
Okay, thank you.
ഹലോ നന്നായിരുന്നു നല്ല ഇതായിരുന്നു എസ്പെഷ്യലി ഫോർ PG ആൻഡ് റിസർച്ച് സ്റ്റുഡൻസ് ആൻഡ് ഓൾസോ ഫോർ ഗ്രാജുവേറ്റ് സ്റ്റുഡൻസ്.
Okay. I hope they were able to follow the technique, you know.
Yes, ma'am. The presentation was very clear, dealing from the basics till the advanced level.
Thank you. I tried. Okay. Bye, everyone. Bye.
Ma'am, that was a wonderful lecture, ma'am. Thank you so much.
Thank you, ma'am. Thank you, Sarvanakumari, ma'am, for joining the meet.
Okay, ma'am. Our students also participating, ma'am. Thank you so much.
Yeah,
Bye.
ask them to fill the feedback link, ma'am.
Okay, ma'am. Certainly, ma'am.
Okay.
Okay.